Hello, I'm Mark Esposito, the founder of KayoThera, here to tell you about our novel approach to developing a new cancer immunotherapy. Our company was founded off the unexpected observation that in the mid-1990s, numerous different clinical trial centers hypothesized that retinoid pathway activity would either prevent or treat various different types of solid cancers. However, there was the unexpected finding that almost universally, retinoid pathway activation led to increases in cancer rates in the chemoprevention setting, as well as worse outcomes in the interventional setting. More recently, we've come to appreciate that this observation was due to the fact that the retinoid pathway is almost exclusively responsible for enforcing immune tolerance in the adult human body. Now, the retinoid pathway is that signaling occurs when vitamin A and various different inputs lead to the generation of bioactive retinoic acid, which when it binds to naive T cells, enforces the differentiation of regulatory T cells while blocking the differentiation of effector or inflammatory T cells. While numerous different large pharma organizations have tried to drug the nuclear receptors, they've all failed due to difficult pharmacology. On the other hand, we've identified very specific ALDH enzymes that are responsible for generation of this bioactive enzyme. And through inhibiting these enzymes, we can control the retinoid pathway in the entire adult body. Now, with these compounds that we've developed and our late lead candidates, we are the first company to develop safe, drug-like, specific, and highly potent compounds against this pathway. And so where we're immediately interested in applying these compounds is in soft tissue sarcoma, a rare and intractable cancer type where retinoid pathway activation is relatively ubiquitous. And thus, in preclinical syngeneic models, we've already demonstrated that our compounds are exceptionally potent as oral monotherapies. And even more importantly, in these various different subtypes of soft tissue sarcoma, these are very, very highly aggressive cancers that are also unresponsive to PD-1 while demonstrating very, very high recurrence rates. Thus, we are exceptionally interested in understanding how our compounds combine with anti-PD-1 therapy. Thus, in these fibrosarcoma models, which are normally unresponsive to anti-PD-1s, as shown by the purple here, our compounds already exhibit exceptional monotherapy activity, while combination with anti-PD-1 leads to almost full remission of these established cancers, while demonstrating exceptional tolerance in the animals that we're treating. We aren't only applying this pipeline to these rare and intractable soft tissue sarcomas. We're also interested in expanding to numerous other solid cancer types, both in cancer types where there's no response to PD-1, as well as those that develop acquired resistance to PD-1, such as TNBC, non-small cell lung cancer, and melanoma, where high expression of our targets predicts worse outcomes in these various different solid cancers. Now, we've already identified development candidates for drugging this pathway, with compounds that show sub 10 nanomolar activity in cell assays measuring retinoid activation. They demonstrate exceptional bioavailability, as well as crossing all of our thresholds for plasma half-life, AUC, various different selectivity and toxicology filters. This entire program has been led by various different KOLs, as well as former directors of medicinal chemistry at Boehringer Ingelheim, Merck, and Vitae Pharma, as well as various different drug discovery leaders, cancer biologists, and clinical immunologists. And with that, I'll draw that all together with our summary, which is that we're the first company to develop inhibitors of the retinoid pathway that are orally available and potent as single agent immunotherapies. And furthermore, we intend to apply these to cancers to sensitize them to anti-PD-1. And with that, we're currently raising $10 million to advance this pipeline to first in human trials.